Welcome again in my online session on medicinal chemistry. We'll discuss here a general considerations of the medicinal chemistry. Drug action. So here one should be clear with what is a drug. Every chemical entity is not a drug. The drug is something that is used for the treatment, and it is efficacious as well as a safe. A chemical entity, if it is safe and efficacious, if it is found safe and efficacious for the treatment of any particular disease, then such a chemical entity can be approved as a drug and can be used in the treatment, in the clinical practice. So this is how a drug comes in, and let us see how it acts. Suppose you hit your knee and there is a pain in your knee. You go to the doctor gives you a diclofenac tablet, you get it, and you will find that there is relief in the pain in the knee. How it happens? Is it a magic? No, it's not a magic. There are many things taking place in a body after swallowing that diclofenac tablet till the relieving the pain. Let us see what happens when you swallow a diclofenac tablet. It's a tablet, solid dosage form, which contains a diclofenac as a drug, active pharmaceutical agent, as well as there are some additives which are used to formulate that drug into a tablet. So when such a tablet is swallowed in the stomach, it disintegrates, the tablet disintegrates into the small granules which is containing a drug, that is diclofenac. Then where the drug gets dissolved, diclofenacs get dissolved and it gets absorbed into the systemic circulation, or it gets absorbed into the blood. This is a process of absorption, drug absorption. Once drug is absorbed into the blood, that blood carries this drug to the whole body. Wherever blood reaches, at every places, every cells and tissues of the body, the drug reaches. Okay, then this is called as drug distribution, absorption and distribution. Now there are some organs where drug undergo a metabolism and elimination. There are some enzymes or some reactions, where biochemical reactions, where a drug entity is converted into more polar form so that it can be easily eliminated from the body. So this is a process of pharmacokinetics, that is absorption, distribution, metabolism and elimination. This is what body does to the drug. Okay, now at the same time, what happens that you get relief in the pain, relief from the pain. When this drug is being distributed, it is also reaching to the knee where there is a pain. What is the reason of pain? Because your knee there is some damage in the tissues which has released arachidonic acid. That arachidonic acid is converted into prostaglandins and prostacyclins due to enzyme cyclooxygenase, and such markers are resulting into a pain situation or inflammation situation. Now what happens? This diclofenac, blood carries this diclofenac to the damaged tissues of the knee, where it gets entry into the damaged tissues, where he finds the enzyme diclofenac. It binds to the enzyme diclofenac, that is its target, and it inactivates that enzyme diclofenac so that it stops conversion of arachidonic acid to the prostaglandins and prostacyclins, which are ultimately giving you the pain. So this is how a drug binds to the receptor or its a target or enzyme, that is diclofenac binds with enzyme cyclooxygenase. This is an interaction of a drug or a ligand with the target. This is known as pharmacodynamics. This is what drug does to the body. Okay, so this is how the drug action is observed. When that drug binds to the enzyme cyclooxygenase, it prevents conversion of arachidonic acid to the prostaglandins and prostacyclins. Ultimately, it relieves the pain. Okay, so this is how it happens. Does it mean that immediately after swallowing that diclofenac tablet, there is a relief in the pain? No, because it takes some time. It needs some amount of drug to be reached to the site of action. Some amount of drug should interact with the target enzyme, and then the action will be observed. So this is what is, you can say that, a typical plasma concentration curve here. Now you can see here the drug is absorbed. Drug is absorbed. It reaches to minimum effective concentration. Here you can see minimum effective concentration. Then still its concentration in the blood goes on increasing. This is the C max. You can see this is the C max, where the concentration of drug is highest. Then it starts declining. The concentration of drug starts declining because of the elimination and metabolism, or metabolism followed by elimination. So the time required for a drug to reach to its minimum effective concentration, it is called as onset of action. Then a time at which it shows highest concentration in the blood, it is a C max. And this duration, this is a duration of action. This time is a duration of action. And if the concentration of drug goes above this is minimum toxic concentration. If concentration of drug goes above this minimum toxic level, then it starts showing adverse effect. So it is a care should be taken that the concentration of drug in the blood should be in this desired therapeutic range. So it should be above minimum effective concentration and below this minimum lethal level. So drug will be absorbed, distributed, reaching to the site of action, binds with the receptor or target, and shows biological action. This is what is PK/PD, that is pharmacokinetics and pharmacodynamics of the drug. Moving to the next, what are different types of drugs? Broadly, we can classify drugs as structurally specific drugs and structurally non-specific drugs. So what is structurally specific drug? That structurally specific drugs are those drugs whose action is dependent on its interaction with a target, that is receptor or enzyme, and if a structure of drug is changed, its action changes. Likewise, so the modification in the structure alters its interaction with its target and ultimately alters its action. Whereas structurally non-specific drugs, their action is independent of its structure. The example, well-known example is general anesthetics. If you look at the list of general anesthetics given on the slide, it may be a xenon or N2O or liquid ether or haloethane or some benzodiazepines or some opioids. All these different drugs which are used as a general anesthetics, they all have their different structures, but their action is same, that is general anesthesia. Because its action doesn't depend on its structure, its action depends on its concentration at the site of action. It works on the Ferguson principle. So these are structurally non-specific drugs. Moving to the next. Now how these drugs are developed? The drugs are developed from hit to lead to new chemical entity. or a drug like moiety or a drug, right? So a lead is a compound which has desired biological activity, but along with it has some undesirable side effects, or it has undesirable side effects that cannot be acceptable. So these side effects needs to be reduced. So the lead needs to be optimized so as it shows more activity, more efficacy and less toxicity, so that it can convert into the drug. So how this lead were found, how to search lead? There are various methods by which lead can be found. That may be a serendipity or random screening, non-random screening, metabolite studies, clinical observations, or a rational drug design. Let us see how these methods were useful up till now, or in future also, how these methods could contribute in the drug design. The well-known example of serendipity is discovery of penicillin. What is serendipity? It is accidental discovery. The penicillin was discovered accidentally. The biologist or microbiology scientist Alexander Fleming, he was working on a microorganism strains like Staphylococcus aureus in his lab. So one fine day he decided to go on vacation, so he closed his lab and went on vacation. A week after he came in, he opened his lab where he kept there his plates containing Staphylococcus aureus culture. He found that on the plates where he wanted to grow Staphylococcus aureus, there are some regions on the plate where there is no growth. There are some regions on the plate where there is no growth of the Staphylococcus aureus. He tried to identify what is the reason. What is the reason of no growth in particular area? Where he found that there is some contamination on the plate, and because of that contamination or around that contaminant, there is no growth of Staphylococcus aureus. Then he tried to find why. What is the reason? Why around this contaminant there is no growth? So he identified that this contaminant is a fungus. It was a fungus known as Penicillium chrysogenium. And this fungus, it releases secondary metabolite, and that secondary metabolite released by that fungus is antimicrobial, so no growth. And then after he identified the released secondary metabolite from that fungus Penicillium notatum or Penicillium chrysogenium, that was a penicillin, that is penicillin G or benzyl penicillin. This is how the penicillin was accidentally discovered. Now, when this penicillin was discovered, it was given by a parenteral route. If you look at the chemistry of penicillin, then you can see that it contains this beta-lactam ring, 4-membered beta-lactam ring. This 4-membered beta-lactam ring is fused with 5-membered thiazolidine ring, and this total is called as a Panum ring system. To this Panum ring system, at 6th position, acyl amino side chain is connected. At 3rd position, there is a carboxylic acid group, and this is intact molecule, or this is intact nucleus of the penicillin type of antibiotics. Now this penicillin G, a natural penicillin G, was inactive by oral route. When it was discovered, it was required to be administered by injectable route, because if it is given by oral route, it was found inactive. Then he tried to find a reason why it was inactive, where it has been found that this penicillin, a natural penicillin, to show its antimicrobial activity, because it is inhibitor of enzyme peptidoglycan. which is involved in cell wall synthesis of microorganisms, right? So this enzyme transpeptidase inhibitor, penicillin, it needs intact beta-lactam ring to show its biological activity. If anything that breaks this intact beta-lactam ring, penicillin becomes inactive. And this beta-lactam ring can be damaged in acidic condition and also in a basic condition. In acidic condition, either it rearranges, it changes, or it opens to generate other degradation products like phenylic acid, penicillamine, phenaldic acid, or penicillaldehyde. Such a conversion, chemical conversion of penicillin leads to its inactivation. Or in a basic condition, it converts into penicilloic acid by opening this beta-lactam ring, and further it may go to the penicilloic acid. So this is how penicillin can be deactivated in acidic as well as basic conditions, so it is orally inactive. The other thing that could inactivate a natural penicillin, that is benzyl penicillin, is enzyme beta-lactamase. This enzyme beta-lactamase is generated by some class of microorganisms. This natural penicillin G is only active against gram-positive bacteria. It is not active against gram-negative strains, because gram-negative bacteria generate enzyme beta-lactamase that inactivate penicillin by acting on this beta-lactam ring of the penicillin. It acts here and it opens the beta-lactam ring. Even a cephalosporin, it is another class of beta-lactam antibiotic. It also gets inactivated due to enzyme beta-lactamase produced by gram-negative strains of bacteria. It opens the beta-lactam ring, generating this inactive metabolites. This is how the benzyl penicillin can be deactivated. So this is what is the limitation of natural penicillin, that is benzyl penicillin. So then after, research moved on to the generation of some semi-synthetic penicillin analogs that could be given by oral route, so that could be stable in acidic as well as alkaline environment, and to generate a broad spectrum penicillin analogs that could be active against gram-positive as well as gram-negative strains of the bacteria. So here the modifications at the side chain has been done, and the ampicillin, amoxicillin, methicillin, cloxacillin, dicloxacillin, likewise various semi-synthetic penicillin analogs were developed. They can be given by oral route. They are stable in acidic as well as basic environment, as well as they are showing activity against gram-positive or gram-negative strains. That is, their spectrum of activity can be improved by structural modifications in the basic penicillin. That is a semi-synthetic one. Now, the another well-known example of serendipity is a discovery of benzodiazepine class of drugs, diazepam. See, this story is of scientist Leo. He was working in a Roche laboratory, and he was working on a project where he synthesized around 242 molecules, and he screened these 242 molecules for various activities. Now, after screening, he found that none of the compound is active, so he decided to terminate the project. During cleaning of the laboratory, after 2 years, during cleaning of the laboratory, he found that there is one vial which is untested. So he decided to test this compound which is untested in order to complete the formality of the project, and that compound was tested and found a fantastic activity on central nervous system, and it was a librium, that is chlorodizep oxide. Now, then after, In 30 years, this benzodiazepine was discovered. It is benzodiazepine 7-member ring system, which is acting on as a GABAergic agonist. Now all benzodiazepine analogs are there in the market because of accidental finding of chlorodiazepine oxide, because just to complete the formality he decided to test that untested vial which was containing chlorodiazepine oxide. So this is accidental discovery. Now other method like random and non-random screening. Random screening means what? Just have a chemical compound and screen for this activity, that activity, and all this. Okay, if you find something, then you can proceed in this direction. If you don't find anything, forget. Likewise, this is a random. It is a time consuming one, no rational. So in order to reduce some time in the random screening, the other way they found that non-random screening. Suppose you have 100 compounds, different compounds, then group them according to their structures. These 10 compounds have some structural similarity, other 10 have some different structural similarity, and instead of screening all 100, one can screen the representative of a particular chemical class, right? And whether that representative compound shows any activity, then screen in detail about the different compounds from the same class. Likewise, so this is a non-random screening. So by this way, random screening and non-random screening, the well-known antimicrobials or antibiotics like streptomycin and tetracyclines were discovered. Now the tetracycline also have some chemical instability, you can say. The tetracyclines, if you look at the ring, it is, there are 4 rings, 4 fused rings, so it is a tetracycline. Out of that, ring A is aromatic and the rest are non-aromatic. Okay, look at the structure of tetracycline. This is one ring. Out of these 4 rings, one is aromatic and others are not. Now this is a minimum structural requirement for tetracyclines to show antimicrobial effect. This tetracycline in acidic condition or in a basic condition alters its basic structure. If you look at this, in acidic or in basic condition, there is alteration in this original tetracycline molecule into a different one, and because of that it inactivates the tetracycline. So in order to improve its stability and in order to improve its efficacy, the tetracycline molecule, if you look at this, they were structurally modified into chlorotetracycline or oxytetracycline, doxycycline and all other tetracycline analogs by making alterations at here. By making alterations at here to give some different stable molecules which could be better active than original tetracycline molecule. Now the other way by which the drug molecules or a lead could be identified, that is metabolite studies. You know that drug metabolism means drug will be converted into some other chemical entity, preferably it is more polar so that it can be eliminated from the body. Sometimes metabolites are better active than the original one. See, on this basis the sulfonamide class were developed. The well-known scientist Gerald Domark, he was working on a dye, azo dye known as Prontosil, and he wanted to test this azo dye for its antimicrobial activity. He tested azo dye for its antimicrobial activity on a petri dish in vitro, out of the body, where he found that azo dye is not showing any antimicrobial effect. But when this azo dye is administered to the living being, then that is in vivo study, then he found that if in vivo studies this azo dye shows antimicrobial effect. He was surprised. He tried to find what is the reason. Outside the body, in vitro studies, it is inactive, but inside the body it shows antimicrobial activity. He tried to find the reason, where he found that in the body this prontosil breaks here at azo linkage, generating sulfanilamide, para-amino sulfanilamide, and this shows antimicrobial effect. Now this sulfanilamide is a lead molecule in development of sulfonamide class of drugs. The sulfonamide, it has antimicrobial effect, but it is toxic. So sulfanilamide itself cannot be a drug. But the structural modifications in sulfonamides were done to generate various different class of sulfonamides like sulfamethoxazole, sulfadiazine, sulfamarrazine, and so on, that are in use as sulfonamide class of antimicrobials. The another well-known example of this metabolite study is a development of fexofenadine. The fexofenadine is a metabolite of terfenadine. The terfenadine is antihistaminic. It is used in allergic conditions, and it prevents the release of histamine. Okay, so it doesn't interact with any histamine receptor, but it prevents release of histamine. So it is not giving sedation like side effects like other H1 blockers. But while during the study it has been found that this terfenadine converts into fexofenadine by oxidation of this methyl group, and this one, its metabolite is more active than previous terfenadine. So nowadays this fexofenadine is used as antihistaminic, which is better, much better than terfenadine, earlier used one. Now because we talked about sulfonamide, let us see how the sulfonamide acts. The sulfonamides are competitors of PABA in microorganisms. So see, the folate coenzymes are required for any living cell for its multiplication, microbial cell as well as human cell. Human cells get folic acid from diet, and that folic acid will be converted into folate coenzymes, and that human cells can use. But in bacteria, the dietary folic acid is not permissible in the bacterial cell. So bacteria, they have to find their own way to synthesize folic acid and folate coenzymes. So bacteria generate their folate coenzymes from dihydropterin pyrophosphate and PABA. These two binds to this DHPase enzymes, generate DHP, and from this DHP the dihydrofolic acid and tetrahydrofolic acid like folate coenzymes are synthesized. Here sulfonamides, being structurally similar to PABA, they bind at a binding pocket of DHPase in place of PABA, leading to faulty folate coenzyme synthesis, leading to stopping multiplication of the microorganisms. So it shows bacteriostatic action. Another way is a clinical observation. How clinically? By clinical observation drugs could be developed. See, you must have heard about a drug Sildenafil, that is Viagra, which is used in impotence condition. So how it was developed? Earlier the Sildenafil drug was under clinical trials for its anti-anginal effect, angina pectoris, that is constriction of coronary arteries, so pain in the chest. So this Sildenafil was under clinical trials for the dilation of coronary arteries. But and it shows the dilation of coronary arteries because of it is inhibitor of enzyme phosphodiesterase. If this phosphodiesterase is inhibited, the cyclic GMP will not be converted to GMP. It will remain there, which will reduce intracellular calcium. So it will prevent constriction of smooth muscles, so smooth muscle relaxation and increase blood flow. This is how Sildenafil acts. But during clinical trials, it has been observed that the Sildenafil is more active in a sex organ than in your heart. So then after it had been decided to use this Sildenafil as in impotence conditions instead of as anti-angina. Another example of clinical observation is a Tolbutamide. It is a class of, you can say this is a class of sulfonamide, and it has been observed that sulfonamide class of drugs shows hypoglycemia as a side effect. So the scientists decided to enhance this hypoglycemia side effect, and they resulted into development of Tolbutamide as an oral hypoglycemic agent, and it has been used as an oral antidiabetic. So this is an observation by which a sulfonamide class of drug can be developed into antidiabetic Tolbutamide class of drug. Now this is what we talked about, how the drugs or leads could be identified and ultimately drugs were developed. But nowadays the rational way of drug design is coming in, and this rational way of drug design will be based upon the interaction of drug or ligand with its target. So if one knows the structure of drug and its target, then we can computationally check whether such kind of interaction in the body is possible or not. So how this is done, this computer aided drug design, for that one should know either structure of target or structure of ligand or both. Suppose if we know the structure of protein or target and structure of ligand, in that case we can go for structure based drug design like docking and all. This can be done by which the drugs can be developed. Okay, if suppose you don't know the structure of ligand, but if you have a structure of target, we can go by de novo drug design. Means this is like, you know, the finding a key. If you have a lock and key model, see, lock fits, key fits into the lock and it opens. If their levers are matching properly, then only that key can fit. Now here, if you know the key, if you know the lock, then you can develop a duplicate key. It's easy way, right? Here in this case, if you imagine that lock and key, here you don't have a key, you just have a lock. So from the internal structure of that lock, the internal levers of that lock, we have to generate a second key that could open the lock. This is what is a de novo design. Then here, if a structure of target is not known, means a structure of lock, you don't have a lock, but you want to develop a duplicate key from existing key. So this is what is a ligand based drug design, a QSAR or pharmacophore. That is what is a development of duplicate key from existing one without considering the lock. And if you don't have both a structure of target as well as structure of ligand, in that case we can't go for a computer aided drug design or a rational way of drug design. So this is what is a general fundamentals of medicinal chemistry. This is what is how drugs were developed and how they could act. Thank you.